This lecture covers alpha and beta diversity and detecting differences among treatments using generalized linear models and simple analysis of variance. Recall that we sampled 3 transects in an area that had been logged and 3 transects in an area that had been unlogged, using 1 meter squared frames in each along the transect lines. The goal of the project is to determine if plant species composition still differs 50 years after logging in the logged area, or has it recovered to similar levels and composition as the unlogged area. So the homework has 2 basic problems, and that's: are there differences in alpha diversity between the logged and unlogged plots? And the second question is, does plant species composition differ between the logged and unlogged treatments? There's 2 sub-questions for each of the main 2 questions. And the first, when we're working on alpha diversity, is does species richness differ between the treatments? And the second is, does the diversity measured by Simpson or the Shannon index differ between the logged and unlogged treatments? The 2 sub-questions associated with the beta diversity differences, does plant species composition differ between logged and unlogged areas, are plots more similar within logged areas compared to between logged areas, and what species had similar cover between treatments? So for your homework, please write a brief introduction about our project and about disturbance in northern Rocky Mountain conifer forests. And then write a brief method section that includes a data analysis section. So less important to me in this section is describing how we sampled, but instead describing how we conducted our statistical analysis. So then add a results section structured based on the questions above. And then finally, write a short, concise, but informative discussion that concentrates on the ecology, not the statistics. I want to hear about the biodiversity differences and the results that we got, and then put those in context of the question, does plant species composition still differ between logged and unlogged areas 50 years after logging? So the script and the homework are loaded up on D2L. And so I'll just go open the script right now, set my working directory, and get all that in order. So here's the script for the homework. Our console, you can see I opened it up, and we have the working directory set correctly up here at the top. And now we'll go over to the actual R script that contains the analysis of the data. So the data is called hi.dat, or for highlight, because that's where we were. And the file is called treatment_data_2015.txt, and it is a tab delimited text file. So I'll just read that in like we always have. No errors, which is good. And then I'll check out, just start looking around at the data, the first thing you usually do. You can see we have, these are the names of our columns, so species as columns, and our rows are plots. And so what I usually do when I make these sorts of when I do these sorts of analyses is I separate and create an environmental data frame and a vegetation data frame, because we really want to be able to calculate species richness, alpha diversity, and beta diversity metrics using just the vegetation data, so just the columns of species, and separate out the environmental data and use that as our independent or predictor variables. So I did that right here by separating these out, and you guys can look at this information. You can go through the code and look, but basically I looked at the names, and I know that I want columns 1 through 7 because that includes our environmental data. So 7, this would be 7 right here. So 5, 6, 7. So 1 through 7 is our environmental data, including canopy cover, amount of rock, litter, bare ground, the location, the transect, and the treatment. So treatment will be our most important variable going forward. And so I subset that out by just doing it this way. Hi.dat, I want all the rows because I want all plots, and I want columns 1 through column 7. So I can run that code quickly. See that now the names of just the E and V data set are treatment, transect, location, bare ground, rock, litter, and canopy cover. I'm going to do the same for the vegetation data, first by checking the dimensions. So we had 30 plots, as you'll remember, and we recorded 44 species. So I'm going to take then columns 8 through 44 and make that the vegetation data. And we can call it the names, just check. You can see we start with our Abies laciocarpa and end with Viola orbiculata. And so we have all our species here. And now we need to move on to answer questions at the scale of the alpha diversity level. So we want to calculate richness, the number of species in a plot, and we'd like to calculate the richness evenness index, or the diversity indexes, Shannon and Simpson. And see the lecture also posted on D2L about Simpson's and Shannon and Simpson's diversity indices. So we'll just skip down here to the first sub-question. Are there differences in plant species richness between treatments? So the first thing we need to do is calculate our richness, and we've done this a couple times in class. Using this sign command, we're going to convert the whole vegetation matrix into values of 1 or 0, because we want to add up. If the species occurred in the plot, we want it to get a 1, and if it didn't occur, a 0. So we're using this sign command like we did in the presence absence homework last week. And you can always type $sign to see exactly what's going on. And so we're going to take this matrix of presence and absence now and sum across rows, because rows are our individual plots. So here we're summing across rows using the row sum command, and there's lots of different ways you can do this. But just simply, we're just going to sum across rows using the presence absence. So every time a species occurred in a plot, it gets a 1. So we'll add all those up and we'll get our richness values. You can see them there. And if we wanted to just check, we'll check the length of rich. We could see that it's 30, so we know it's the right length, 30 plots, 1 value for each plot. And now we can start to explore whether there's a difference between logged and unlogged. And we'll do that using our environmental data set and the column treatment, so logged versus unlogged. And we can make a box plot of richness, and we can just run that quickly. You can see there we get the results, and we can start to assess whether we think there's a significant statistical difference or not. Recall the outlying whiskers are 95% of the data, the box is 50% of the data, and the heavy weighted line through the middle is the median. So now let's think about testing that statistically to get a p-value to find out if there's a relationship between treatment and the amount of richness we observe. So we're going to make a generalized linear model of richness in response to treatment, and we'll use the Poisson distribution just like we did last week with our count data. And this is again count data because we're counting the number of species that show up. And we'll run the model, and it worked fine, and we can call up the summary command if we're interested in the coefficient estimates and some of the p-values. And you can see that here we have our model summary, just like I highlighted in the regression lecture, also on D2L. And so we know where to find the coefficient estimates and the p-value associated with the test of whether there's a non-zero relationship between the differences in the mean richness in the logged and unlogged area. And so you would get some output right there, talks about and then shows the p-value for the the treatment. And then we can do that in another way. We can think of using the likelihood ratio test, or one-way analysis of variance here. And the likelihood ratio test basically calculates the probability that the factor you entered into the model is significantly improves the model or not. And it's done based on the ratio of deviance, or the residuals here. So does the ratio of the residuals explained to the total amount of the residual in the model significantly improve, and we derive the p-value using the chi-square distribution. It's called the likelihood ratio test, the goodness of fit test. Those are the two most common ways it's referred to. So here we're just giving to the ANOVA command, the analysis of variance command, our mod, which is our model from the line above, and we're using test equals chi here because we're doing the chi-square goodness of fit test or likelihood ratio test. So we can run that, and we get out our analysis of deviance table, because it's called deviance in generalized linear models rather than the residuals, because we'll not worry about why. But basically, we'll say if we had a null model here, a generalized linear model that was fit with only an intercept term, and then we added the treatment variable to that term, did we predict more of the deviance? So the deviance predicted was accounted for by treatment was only 0.22698, and the total deviance of the model was 18.24. And if we subtract 18.24 from this, we'll get a residual deviance. So what's left over of 18.01. And we'll use that, think of this ratio here. Did we significantly explain a lot more of the deviance? And the answer is, you figure it out. But basically, we'll derive this p-value, which is the same one you'll note from above, using the chi-squared likelihood ratio test. So that helps us answer, does richness differ? But we have this other confounding problem that other variables, like the amount of canopy cover, may vary between treatments and affect the understory plant community. So we can just quickly test to see if we had significant differences in canopy cover between the different treatments, logged and unlogged. And we're essentially doing the same thing. The syntax I wrote a little bit different here. So we're saying I told it to take the data, this environmental data set, and look for the column name canopy and look for treatment, and I want canopy as a function of treatment for the box plot. So then that's what we get. So you guys should add in the X and Y labels. And then we make a generalized linear model in the same fashion as we did the last model. This time we don't need to specify the Poisson distribution because we don't have this integer count data. We have continuous variables between 0 and 100. And we can run that. We can run the analysis of variance test again, and you can see whether canopy cover had a significant effect on treatment. And remember, we're hoping that if we have a significant effect, the deviance explained by treatment will be really high, and it'll reduce the residual deviance by a whole lot, and then ultimately affect the p-value based on the likelihood, based on the ratio of residual deviance to null deviance here. So I hope that makes some sense, but that's basically what's going on with the simple analysis of variance test. So further down in the script for beta diversity and also for what's just below alpha diversity, we are going to need our library vegan again, so we can load or require library vegan. And now let's go down and let's calculate some diversity indices. So alpha diversity indices account for both evenness, how even the abundance of a species are in the community. So if a community's really even or a plot's really even, all the species have similar amount of cover, density, or frequency. And if it's uneven and skewed, then one species is really abundant and the others are rare. So these are our alpha diversity indices. The two most commonly used are the Simpson and Shannon indices, and they basically sum up the proportion of the community that each species is. And see the lecture on alpha diversity indices on D2L. So we're using this function out of the vegan package called diversity, and I can go over and have a look at vegan or at diversity just to see what. So remember, always question mark, and the function you're interested in to call up the help in R. And you can see that it specifies what the usage is. So the way we're entering it is X will be our community data, our veg data frame. And we're going to specify the index here, either Shannon, Simpson, or inverse Simpson. And then you can make some other changes which we don't really need to go over. We're using mostly the standard package. First, we'll calculate the inverse of Simpson and the inverse of Shannon. And those went well, no errors. But then we might want to actually then compare that to our richness values. So richness is one of our alpha diversity measures, and these two are the other alpha diversity measures we're looking at. But are they correlated is the question. So first we can call up dsim, which is our Simpsons, inverse Simpsons index, and we can see we get a whole bunch of numbers here. And so the higher the number, the more diverse a plot is. And so one question we might ask ourselves, are these diversity indices that account for evenness and richness independent of our just richness variable? So if we were interested in finding out, we can just type core for correlation, and we could type dsim and rich. And when they're both 30 rows long, or now they're vectors of length 30, and they have the same order, so we can compare the plot level richness. And we can see that the inverse Simpson is positively correlated by 0.36 or 36% of our but that's actually fairly independent, I would say. The cutoff usually for saying something is not, that two variables are not independent of each other, is somewhere around 70% if you were using them as both predictor variables in the same model. And so we can run that for Shannon too. I'll just type that, Shan. You can see that the Shannon index is slightly more correlated, up around 50%, but still fairly even. So we're just repeating the same things that we did above, and that's just to make linear models and box plots of the similarity in response to the treatment. So here I wrote the code a little bit different. I just nested the GLM formula and function call inside of the analysis of variance and didn't save it as an individual object. We'll run the same amount of code and it all works. We get some p-values and some analysis of variance tables, including some graphics, and I'll leave that up to everyone to go through themselves and find out why, or find out if they significantly differed or not. And I'm going to move on to talking about beta diversity now. So there's also a beta diversity lecture that will appear on Friday on D2L that will cover how these dissimilarity measures are calculated. But basically we are comparing how similar these plant communities and how similar plots are to each other using an index. And in this one we're going to use the index, the Bray-Curtis index, which we're going to calculate the vegetation distance. VegDist is the command, and we'll give it our matrix of vegetation data, and we're telling it to use the Bray-Curtis index. Now we'll go over the Bray-Curtis index in the other lecture, and we'll go over some other dissimilarity indices like Jacquards or Sorensen's in the lecture on Friday too. But basically we're calculating how similar is each plot to every other plot. So we have 30 plots, and 30 plots times n minus 1 combinations will give you the size of the data matrix. So if we just run these commands quickly, the VegDist command, we get the VDist, and you see we get this triangular looking matrix up here. And because the matrix is a mirror image of itself and the diagonals are all comparing site 1 to site 1, and site 2 then to site 2, and site 3 to site 3, forming the diagonal of the matrix, then comparing site 1 to site 2 will give you the same answer as if you compared site 2 to site 1. So we end up with only half a matrix that's unique. So we'll just we hold on to that half unique matrix here. So for example, the similarity of plot 6 to plot 1 was 0.66. The similarity of plot 7 to plot 2 is 0.57. So we're going to use all this information to calculate whether there's dissimilarity between treatments. So you can imagine when we calculate a big dissimilarity matrix like this, that we can compare within treatments to among treatments as well. So there's a nice command in vegan called mean_dist that will calculate the mean distance between the treatments and within treatments. So here I ran the mean_dist command from my script, and so when we compare logs to logged, the dissimilarity between the two is 0.77, and between logged and old growth it's 0.79, and old growth to old growth is 0.70. So the highest values of dissimilarity are between old growth and logged, and see how this one matches this one because we have the symmetric matrix and only a certain number of unique positions, and that we have 15 plots in each. It gives us our nice number grouping, and so the within similarity is highest between old growth and old growth, then logged and logged, and the highest dissimilarity is between the old growth and the logged. So hope that makes some intuitive sense to everyone. So now we can use that information. Well, first we'll calculate the Jacquards, the same using the Jacquard index. And I used this one in the lecture. It's quite an easy one to understand, where it's A over A plus B plus C, where A is the species shared between a pair of plots, B is the unique pool and unique species to pool B, plot B, and the C is the number of unique species unique to plot C. So we can also calculate this VegDist Jacquard, and we can bring up the same information. And we can see the numbers are qualitatively pretty much the same. The log to logged actually has the second ranked. So the ranking of how they're dissimilar to each other is the same. The lowest dissimilarity is between old growth and old growth plots. This next is between logged and logged plots, and the highest difference in beta diversity is between logged and old growth plots. So now we need to figure out, does that translate into a significant difference? So we can use this function Adonis, and Adonis is quite a handy function. We'll call up the help. And then Adonis is permutational multivariate analysis of variance using distance matrices. And I'll let you guys read through some of this, but it's related to MANOVA, multivariate analysis of variance. And it's permutational, so it does it on a simulation basis by permutating all the values. So we can just give it the same format, or a format similar to the GLM command, where we're saying take the vegetation dissimilarity from the Bray-Curtis one above and test this in response to the treatment. And so we run that command, and you'll see we'll get this value. So now you can see we have our treatment has a significant non-zero effect. And now our R squared, or our amount of deviance explained, or the is 0.1%. So we're explaining about 10% of the total amount of variation in the data set. 89% remains unexplained, and the total equals 1. So you can see here's these sums of squares again. So we have the 0.92, similar to the other analysis of variance table that we had, where it said here just deviance instead of sums of squares. And the residuals here is what's left over, and then this equals the total amount of the sums of squares, so this total amount of deviance in the data set. So it looks like we have a significant difference between the two. And now we're going to use a technique called principal components analysis to visually plot this data and look at the effect between the two. So principal components, and there are some D2L help videos up about principal components analysis, is a way of putting all this information into multivariate space, and then fitting a plane through that multivariate space so that we maximize the amount of variation we get out of the cloud of the data points, if that makes any sense. See D2L under semester lectures for some of the introduction into calculating principal components. But we're going to let R do most of this for us, and we're going to use this command right now called beta dispersion, or beta disper, and it calculates the dispersion using our vegetation data and our treatment. So we're going to just, and this function is in the vegan library. So we ran this h-p.pco, so our principal components ordination, principal coordinates analysis, and then we can plot that really quickly. And we see you get these points. So the red dot here in the center is our centroid, or our sort of mean condition. And this is the centroid for our other treatment. The blue lines stretch out to all of the points that we have in the data set and correspond to the treatment. So circled for one level of the treatment, triangles for the other level of the treatment, and then this hull is drawn around it, or sort of the capture of the space. This is where the two variables, or this is where the two treatments vary. So there is some overlap, and there's some non-overlap between the two sites. So that helps us think about it. And so R just does the beta dispersion command plots it very nicely for us, but maybe we'd like to see some more about what the attributes are in that high.pco object. So it's a beta dispersion parameter, so we can call it up. And you can see that it gave us not a data frame or a type of names, but a list of what's in there. And so we have the eigenvectors, we have other vectors, distances, groups, the centroids, which are the red dots I talked about, and the actual formula call. And the class of this object is a beta dispersed object. And that the method we used was Bray, and the type was the median. And so now we might like to plot some of this information out, give it different symbols, add some treatment labels to it. So we'll start working through it this way. And all these plot commands can be quite useful for analyzing your own data. So the first thing we're going to do is take this PCO data and go into the vectors. And those are the principal components. So these are the vectors associated with the principal components. And we're going to take number 1 and number 2. So we're going to take PCA axis number 1 and number 2. PCA axis 1 was fit through to maximize the surface, to maximize how many points in the area that this vector passed through the cloud. And then 2 is one that's perpendicular to it. And I'm going to do the PCH, which is the point characters. I'm going to make those values of the treatment. So if we call up just E and V treatment, we get this logged, unlogged. And if I just tell it to make it a numeric value, then it will take the factor and convert it to a numeric value, 1s and 2s. So it's going to plot every plot with 1s and 2s, and we can call that up. You can see we got our circles and our triangles just like in that other graphic. And then we can add in the centroids out of that, just as we took earlier. So we'll take this centroids and, oh, let's see. Oh, didn't do the right line. Here we go, we got the centroids, and we added our centroids, so green for one of them and red for the other. And then we can add in their names. And you can hide, so if you wanted to know, for example, what does high centroids look like, you can just subset and call that up. And you can see we have principal component number 1 and principal component number 2. It goes all the way out to 29, right? Because we had 29 or 30 species. We have n-1 principal components here. So 29 because we had 20 n-1 because we had 29. We have values for every single plot. So n-1 plots. And so we can add that text. I'm using this. So here I made the plotting window, and once the plotting graphic window's open, then I can use things like points. And we gave it the points. We can just run that again and see, okay, we have the centroid points now, and we can add in text. So I added in text near. So what I did was I just looked at the graphic and I said, take the centroid and use the x value and then use the y value, but subtract 0.05 on it so it's not right over the top of the 2 points. And then we can call up and see that we get our logged and unlogged. The centroids do differ from each other, so we do have differences in beta diversity, so differences in plant species composition between the 2 treatments. You can also do that this way and add in logged and unlogged in the same way, just to do it another different way. And so now we know that we have differences in species composition between the logged and unlogged. And maybe we'd like to know what exactly those species are. So what I'm going to do is separate the vegetation data frame into logged and unlogged plots. And I'm basically saying take vegetation and subset it based on treatment, and I only want the ones where it equals log, and then the other one will be the old growth or the unlogged area. So I called those log and unlogged to denote the 2 objects. And so now the 2 data sets are separated, or are subset from each other. We can look at log, for example, and we can see our logged plots. And if we type dim log, we'll see we have 15 plots and 37 species. Oh, but I think this, yep. And then what we're going to do is calculate the mean cover for each species in the logged and unlogged area. So to calculate that mean, remember that our rows are plots, and a species is a column. So if I wanted to find out the mean cover of that species in a treatment, I can take each individual column and calculate the mean across all those of the 15 plots. And so I do that by saying apply to log by 2, and 2 means to do it column-wise instead of row-wise. If I put 1 in there, it would do it row-wise, and it would calculate some value. And I'm saying take the mean of each one of the columns. And if you need to see apply, remember a question mark apply to remember the syntax. And somewhere in here it will say, here you go, for a matrix, 1 indicates rows and 2 indicates columns. You can also give it a 1 and a 2 together to indicate rows and columns. Okay, so I'm saying take the mean of each column for this and call it imcov.l. So the mean cover in the logged and the mean cover in the unlogged. Now we can call those up and have a quick look at them to make sure they look right. So there's the mean cover of all the species in the logged area. So they're not sorted by anything, maybe alphabetically sorted. And we can compare that to the unlogged. But then you end up with these kind of funky looking vectors, and they're all right. They're not too bad to compare, but you have to like really look across the lines. But one thing we can do is we can bind them together using this command rbind. So that'll bind the two vectors together, and we can just run that command. And there we get, yeah, so in the logged, Abies laciocarpa had on average 4% cover, and in the unlogged, 10% cover. Okay, and we can look through all these. But if we'd like to make some nicer tables or really sort out our data, we can do some different things. One easy way to view major differences would be to just subtract the cover of the logged to the unlogged. And because they're in the right order, it's quite easy. We can just look and see now. Okay, now we have one single value. So we know that in the logged, so we're subtracting the second from the first, so there's 6% less cover of Abies laciocarpa in the unlogged as to the logged. So we can think we have that difference value now. We subtracted the unlogged from the logged to look at the difference in means. Another way we can do it is to actually C-bind it together so we get a nice table of it going up and down. And then just to make it look nice, I gave it this sequence. And I told it, and I'm telling it here, bind this vector of the differences right there to this sequence of numbers, but I don't know how long it is, and I'm too lazy to go look. So I say make a sequence from 1 through the length of the difference of b. So that's what that is doing there. And so we can see what the length diff b is by clicking 37, all right. And then we can tell it to make a sequence of 1 to 37 then. And bind those two together. And so we saved that. Now let's go back and we'll look. So now we can see what the percentage difference is between them, and they're in a nice column there. We can look. But then it might also be nice to order these from high to low so we don't have to like search through and really think about it. So what we're doing here is just taking the pdiff, which I named up here for the percent differences, and we're going to order percent differences of the whole data set of all the rows based on pdiff column 1, which is the difference in percent cover. So if we just order pdiff like that, we get a nice difference that's sorted. And so let's think about this. So we have Vaccinium scoparium, which is huckleberry, so ericaceous shrub, blueberries, related to blueberries, same genus as blueberries. And we notice that there's 15% less cover of Vaccinium scoparium in the logged area as compared to the unlogged. So there's less Vaccinium scoparium in the unlogged area. And we can always go back up and see what those actual values were. We'll find that in here. Call it up, let's see. So Vaccinium membranicum, how word? Vaccinium scoparium. Oh, the whortleberry. So in the logged, it had an average of 7% cover, but in the unlogged area, an average of 23.6% cover. So that gives you all the information you need to answer the homework questions and write it up. Hope you guys enjoyed the project. I really did, and I didn't know what answers we were going to get, but we got some quite interesting answers, and I look forward to reading everyone's discussion. So thank you for listening, and thanks for taking the class.